So can you see my screen?
Yeah, that looks great.
Okay, great. So first of all, thank you very much for the invitation. And also thanks to organizers for putting together such an amazing meeting. So we all know that microbes and more broadly microbial communities play important roles in the environment, in industry and health. And their roles can shift from positive, such as providing essential nutrients to their host, to negative, such as causing infections and making their host sick. And so this raises the question of to what extent can we predict how microbial communities assemble, how they evolve, and how they respond to perturbations. But to be able to make such predictions, we first need to understand what is predictable, what isn't, and why. As answering these questions is really important to efficiently manipulate and design microbial communities that perform desired functions. So multiple studies in a range of natural environments have shown that the metagenomic structure of microbial communities across similar habitats is quantitatively convergent, which is illustrated here for plants and for humans. And this convergence at the metabolic level occurs despite a high taxonomic variability as shown here in the bottom panel. And so what this suggests is that the microbiome becomes more predictable at higher levels of organization, but then is more variable at lower levels of organization. But while the metabolic structure may be predictable, actually predicting it would require us to know a lot about these habitats. So know about their history, about their dispersal pattern, and also about their temporal and spatial variations in nutrient and temperature and etc. And really most often we don't have that information, and especially for communities in natural environments. So to address this challenge, our approach in the lab is to use simple model systems where we can control and manipulate the environment in a more predictive manner. And so basically what we do is to watch microbial communities self-assemble in well-defined and controlled environments. And so in a nutshell, what we do, so we first collect a sample from the natural environment, for instance, from the soil, then we bring it to the lab, and then we inoculate a sample which contains a highly diverse species pool into well-defined minimal media, which is supplemented with a single carbon source. And then we let it grow for 2 days, and then we come back and dilute a small fraction into fresh media. And then we repeat this growth dilution cycle multiple times. And then at the end, so once communities have reached equilibrium, we assess the taxonomic composition using 16S sequencing. And all this is done under well-controlled lab conditions. And so then the first question we ask is, so does community assembly in these synthetic environments recapitulate the patterns observed in the natural environments? And so what I'll show you next is the result of an experiment started with 12 different soil or plant species pool and inoculate into fresh minimal media with glucose as the single carbon source, and then passage to fresh media every 48 hours for 12 transfers. And there are 8 replicates for each inoculum. And so shown here is the family level composition after 12 transfers for the 96 communities. So 12 inoculate times 8 replicates. And what we can clearly see is that there is a strong convergence in community composition at the family level, regardless of the initial inoculum. And also most communities adopt similar ratios of the 2 dominant families, so the Enterobacteriaceae and Pseudomonas family. And this occurs despite a substantial variability at lower levels of taxonomic organization, such as the genus level shown here, and even for samples that were inoculated from the same species pool. And so this shows us that with experiments done in these simple and synthetic environments, we observe similar patterns as the ones observed in the natural and more complex environments I showed you before. And so with these observations, we can now ask, so why do we see these patterns of convergence and variability at different levels of community organization? And so first, so why do we see convergence? So essentially, by looking at the consumption and secretion profile of a library of strains that were isolated from the self-assembled communities, we recently showed that this convergence at higher levels of community composition reflects a metabolic self-organization between two functional groups. So between glucose specialists that ferment the glucose and organic acid specialists that cross-feed on the byproducts of fermentations. So they are respiral. And also, what is important here is that these metabolic traits are conserved at the family level, which then explains why we see convergence at the family level. But really, the main question I want to address in this talk is, so why do we see such variability at lower levels of organization, even in replicate habitats? And also, what causes the taxonomic variability within each functional metabolic group? So to address this question, I started a new experiment, but this time with 92 replicate communities, so all started from the same inoculum. And then I propagated them in the same growth conditions as before for 18 transfers. And so after 18 transfers, what we find is that most communities assembled into the metabolic structure I just described, so consisting of fermenters and organic acid specialists. And at the genus level, we again find multiple alternative states in community composition. And also the main difference between these states is the composition of the organic acid specialist group. So while within the fermenter group, all communities contain the same Klebsiella strain, so shown here in dark blue, and that I will call K, within the respire group, we find that communities contain either Pseudomonas or Emergence as the dominant organic acid specialist. And also, what is interesting here is that the identity of the organic acid specialist in turn shapes whatever taxa may be present in the community, which then suggests that the dominant organic acid specialist drives the taxonomic variability observed. So now the question is, so what ecological mechanisms govern whether a community will contain Pseudomonas or Emergence as their dominant organic acid specialist? And so the first hypothesis is random sampling. So the idea is very simple. Because communities assembled in a closed system, so without immigration, any community where Emergence or Pseudomonas was not inoculated initially, so from the initial species pool, then those communities will also not have those strains at the end of the experiment. But when we plotted the abundance of Pseudomonas against the abundance of Emergence at transfer 18, and so here each dot represents a different community, what we find is that in many of the A-dominated communities, P is still there, but it's rare, so it is at less than 1%. And similarly, in many of the P-dominated communities, A is still there, but it is rare. And we also identified a third state where they are both rare. And so this suggests that the alternative states we observe are generally not caused by random sampling from the initial species pool, and they are also not due to extinction of those taxa during the passaging. And so then we go back to the same question. So why don't we observe a state where both A and P are abundant? And so the second hypothesis is that A and P may mutually outcompete each other in a density-dependent manner, which can then give rise to multi-stability. And so to test this second hypothesis, I isolated the three dominant strains, so K, A, and P, and then I grew K with varying initial densities of A and or P, and so in duos or in trios. And then I grew them for 12 transfers under the same conditions as the top-down community assembly experiment. And so with this setup, we can then map a two-dimensional phase portrait with the outcome of competition at different initial density combinations. And so after 12 transfers, what we find is that P and A can coexist with K alone, but not together, which shows that A and P mutually exclude each other. And also importantly here, which species dominates depends on their starting densities. So in the upper left corner of the phase portrait, so where communities were started with high A and low P, then communities converge to a A-dominated state. So A excludes P. And in the bottom right corner, where communities were started with a high P and low A, then communities converge to a P-dominated state, so P excludes A. And finally, then, to confirm that this multi-stability also occurs in the self-assembled communities, I sequenced the full temporal dynamics for a few of the communities. And so here I'm only showing 2 examples, but what we can see is that communities start in the transition region, so where both A and P are at low abundance, but then as communities assemble, they fall into one of the attractors, and once they settle in, then they never switch between states. And so here in the example shown on the left, communities fall into the A-dominated state, so while on the right, communities fall into the P-dominated state. And so in summary, what I showed you today is that for communities assembled in glucose, convergence at higher levels of community organization reflect a metabolic self-organization between 2 functional groups, so glucose specialists and organic acid specialists, and also mutual exclusion between 2 functional taxa can explain the variability observed at lower levels of organization. And with that, I'd just like to thank many people, especially in the Sanchez Lab at Yale, and also collaborators, and thank you. And I'm happy to take any questions.